So we came from R7 or NoSequence division, and I'm going to describe you my ideas and something about research design of my project. So main objective is develop new mapping algorithm. Sometimes people call it also alignment. So what usually people in bioinformatics understand under mapping algorithm? This algorithm for input and just reads, small reads of DNA, DNA in some sequence data, mostly cases some reference genome, and output. For every read we have position in sequence data, sequence genome, probably with some probability or similarity marks. I have some picture. Yeah, the classical usage of mapping algorithms, it's one of the important main steps in sequencing. So we have a lot of reads and we want to map to reference genome and then assembling new sequence from these reads. Okay, there are lots of mapping algorithms with very good performance. A lot of them using FM index and bars that based on Barrows-Will transform, yeah, like Bull-Child. But all of them cannot handle correctly a situation with polymorphic genes. What is polymorphic genes? I give an example. HLA, the human leukocyte antigen, is a very important group of genes that relate to immune system, and there are a lot of alleles of such gene. And at this moment, we know more than 7,000 of alleles, because yes, they shouldn't be polymorphic because we want to have resistance and a lot of viruses. Yeah, and what's the problem with mapping in this situation? If we will use this approach when we try to map to same reference genome, we cannot distinguish these alleles. They will have, yeah, in the result we get the alleles from reference genome. So what we want to do? We want to map not to reference genome, we want to map to some alleles database. We should choose the allele of interest, the gene of interest, and yeah, to provide some database and map reads not to reference to some database. Yeah, this approach significantly increase the complexity of task, because now for every read we have a lot of possibilities there. So it's incredibly huge computation task. And to do this, we are going to use cloud computing. We are going to build a distributed system. Yeah, with hundreds of machines, servers, and transducers. Transducers is very good approach from work for context-based computation. They used a lot in such domains of natural language processing. We have a lot of context things. Okay, and I about something about cloud computing. We are going to use Amazon Web Services. What's Amazon Web Services? It's basically Amazon provides you use their computation capacities, such as servers, database, storages, and key moment of philosophy of Amazon Web Services that you pay only for used resource. You don't need to pay a lot for this configuring. If you want 100 machines, you will pay for 100 machines, not more. Yeah, this example, you can rent 50 high-performance servers for 10 hours and it will cost less than 20 euro. Okay, so now we are working lot with cloud computing and transducers to provide library for work with them. And now I'm going to talk a bit about Nispera. Nispera is my personal project in R7. Nispera is easy but very flexible system, distributed system that for executing arbitrary computation in Amazon Cloud. I will give sample example. You have a lot of amino acid sequences, and you want to use BLAST to determine from which proteins they come, and you should provide this BLAST task in special format, and then you can use Nispera to run it in Amazon, and you can significantly decrease your time, because should be not easy task. Okay, thank you.